Chapter 1, Topic 1.7, Naming, Classifying, and Identifying Microbes. At the end of the lectures for this topic, you should be able to meet the following objectives. Differentiate between the terms nomenclature, taxonomy, and classification. Create a mnemonic device for remembering the taxonomic categories. Correctly write the binomial name for a microorganism. Draw a diagram of the three major domains, and explain the differences between traditional and molecular approaches to taxonomy. For most of the objectives, I've found a lovely crash course on taxonomy, which should help you with most of those. Now, for the other two, specifically, number 3, correctly write the binomial name of microorganisms, and number 5, the difference between the traditional and molecular approaches to taxonomy, I'm going to go into that. So, watch this part of the lecture for objectives 3 and 5, and then watch the crash course on taxonomy for the rest of them. So, binomial naming of microorganisms. We use the Linnaeus system of giving each species a genus and a species name. And the genus name is always capitalized, the species name or the epithet is in lowercase, and both of them are always italicized because they're derived from Latin. So, on an exam, if you had these options, which one would be correct? Okay, see, I'm helping you for the exam. So, these two aren't italicized, so those are out. This one, the genus name is not italicized. Here, the genus name, or excuse me, it's not capitalized. Here, the genus name is capitalized. The species name is lowercase. They're both italicized, so D would be the correct answer. Now, sometimes we abbreviate this, where we abbreviate the genus name to one capital letter and have the species name written out. So, that's binomial naming of microorganisms. That's what you need to know to get this correct on exams and to not have points taken off on your CAP assignments and your oral presentations in lab. Now let's talk about the differences between the traditional and molecular approaches to taxonomy. Traditional taxonomy has focused on differences in shapes of structures and differences in reproductive organs. For example, mammals have mammary glands for producing milk and have uteruses for carrying fetuses. This works wonderfully well until we get to bacteria don't have arms and legs and teeth. So before we developed molecular approaches to taxonomy, we divided them based on shape. Circles or rods or commas or corkscrews. Then we divided them up based on what they ate and what waste products they produced, which indicates what enzymes they have, which indicates also what genes they have, because genes produce enzymes and enzymes produce products. Then we also used the gram stain. Okay, we gram stain organisms, they're either gram positive, gram negative, we go from there. That worked fairly well, but you can see the problems that arose. Think of how accurate we would be if we took a random classroom of people and put all the red-headed humans into one extended family. Then we divided those that produced a urine with a pH of 6.8 into immediate family, and those that produced urine with a pH of 7 into another immediate family. And it's much better if we ask who is related to whom. Well, you can't do that with bacteria. But once we developed molecular biology, basically looking at DNA to determine relatedness, it was like being able to ask the bacteria who was related to whom. And we had to rearrange some of the taxa, but it's all toward making the system more accurate. So in molecular biology, we use DNA sequencing, we look at lipid composition, and etc. Now we still use the traditional methods, starting with gram stain, separating organisms based on shape, etc., but we fit it within the actual relatedness that we've seen from DNA sequencing. Now, we can also use molecular technology to identify relatedness in organisms other than bacteria, but this is microbiology, so that's what we're going to focus on in this class. Okay, so after you watch the crash course on taxonomy, you should be able to differentiate between the terms nomenclature, taxonomy, and classification. Go ahead and create your own mnemonic device for remembering the taxonomic categories. There's all sorts of them out there if you can't think of any. From watching this recorded lecture, you should be able to correctly write and or identify the binomial name for a microorganism, be able to draw 3 major domains, and from this recorded lecture, explain the differences between traditional and molecular approaches to taxonomy.